So hopefully you can all see my screen. Okay, so thanks very much indeed for the invitation to give this talk today. I'm sorry I can't be with you in Dublin. Here in Kingston in Canada, there's about 6 inches of snow and the temperature is about -5. So kind of a normal spring, early spring day, I guess. These are my disclosures. Okay, I'm going to begin this talk with some background information just fairly quickly so that we're all on the same page. What you're looking at here then is the biochemistry, if you like, of haemophilia. This is supposed to represent a membrane surface on cells like platelets. And here in red, we have a protein and an enzyme, activated factor 9. And here in blue, we have the protein that works on, factor 10, and it converts that to activated factor 10. And to do this effectively, it needs factor 8 as a cofactor. And this is a very efficient, effective biological complex that we refer to as the intrinsic 10A's complex. If you have hemophilia B, you're missing the enzyme. So the red protein, the protein that acts on factor 10 is missing, and you can't make this product. And so that's the reason that there's bleeding. You can't go further down the coagulation cascade. And in 1982, we found out about the gene that makes factor 9 from 2 groups in Seattle in the US, Earl Davies group, and from George Brownlee at Oxford. And so this was the start, if you like, of the molecular revolution of hemophilia when the factor 9 gene was cloned by these 2 groups. Now, and here's what the gene looks like in schematic form. It encompasses, it takes up 34,000 nucleotides or building blocks of DNA. And the part that makes the protein is represented by these red oblongs here. And they make the protein. The rest of the things are called introns, which aren't necessary to make the protein. And in total, they amount to 1,400 nucleotides, 1.4 kilobases. And this is what you need for gene therapy. And that's what's being delivered to people who are getting factor 9 gene therapy. So now let's turn to factor 8 for a few slides. So this is what happens when you're missing factor 8. So the cofactor is missing. You can't get the enzyme close enough to factor 10. And so it can't produce this downstream activation protein, factor 10A. And so when factor 8 is missing, this complex is a million-fold less effective. It basically is biologically hopeless. And that's the biochemical basis for hemophilia A. When you're missing factor 8, you can't get the enzyme close enough to the protein it's supposed to be activating. So the factor 8 gene was cloned a year or so after factor 9 in 1984. There were 4 papers in the journal Nature, and they described this gene, which is shown here. So you can immediately see that this is more complicated than factor 9. It also encompasses not 34,000 base pairs, but 184,000 base pairs. So it's much bigger. There are more of these so-called exons, these black bars here. And when you add up the space that all these black bars take up, it comes to 8.5 kilobases, or 8,500 base pairs or nucleotides. And that's what you need to deliver for factor 8 gene therapy. So at this time, back in 1983, I was undertaking postdoctoral studies with Arthur Bloom in Cardiff in Wales. And he wrote this article for Nature in which he described the benefits of cloning genes for clotting factors, that it would help us understand more about how these conditions occurred, how we could diagnose these conditions with molecular genetic techniques. And here's what we're going to be talking about for the next 25 minutes, genetic therapy. So how long did it take to get there? So I've just shown you the genes were cloned back in about 1983, 1984. Within 2 or 3 years, we were using that information for genetic diagnosis. And within a decade, we were making recombinant proteins, which are now being used all over the world. And many of you listening to this call will be treated with recombinant factor 8 or 9 for your conditions. But it wasn't until 2011 when the first clinical gene therapy study really showed to be persistent and positive in humans. So this has taken 25 to 30 years. Now, this slide shows that there are different ways of carrying out gene therapy, and I'm going to go through 3 of these 4 quadrants very quickly. You can deliver genes through so-called non-viral methods, and none of these particular things, and I've listed here some things about nanoparticles and delivering through force into certain organs. These are not even close to clinical development. So I'm not really going to say anything more about this quadrant. Over here in the bottom left, I've listed 3 ways in which you can repair genes or edit genes. And you've all heard, I'm sure, of genetic editing. And those can use strategies like so-called zinc finger nucleases or TALENs. And almost everyone's heard about CRISPR. So these are ways in which you can go into particular regions in the genome, like the clotting factors, and edit the mistakes, edit the mutations, and then make the disease better so you can recover the disease. So this is very interesting, and at some point, maybe in 10 years or so, this may get into the clinic, but it's some way away from that happening. You can also think about cell-based therapies where you take cells from an individual who has hemophilia and then genetically alter those cells outside of the body, so-called ex vivo, and then deliver those back into the donor, the individual who had the condition. This is still being worked on, and ultimately I think will get into the clinic, but not for at least, I think, another 3 to 5 years. And so everything I'm going to talk about relates to viral gene therapy, and I'm going to focus on this now. And before I do that, on the next 4 or 5 slides, I'm going to just give you a little bit of background. Here in Kingston at Queen's University, we have a colony of hemophilic dogs, and this octet here represents one of the most recent litters from that colony. The colony was brought here in 1980 by my predecessor, Alan Giles. The dogs have severe hemophilia A. About a quarter of them have inhibitors to factor 8. They're one of 2 colonies in the world, and we treat them with recombinant canine factor 8. So we make dog factor 8 and infuse these dogs when they have bleeding events. Back in the late 90s, we cloned the dog factor 8 gene, and we subsequently characterized the dog mutation. So we know exactly what the mutation is in these dogs who have severe canine hemophilia A. And then in the middle of all of this, we carried out our initial gene therapy study. So this is now back in 2001, using an adenoviral vector. Not an AAV vector, but an adenoviral vector. And you can see in these 2 dogs, dogs A and B, within 2 days, we took their factor 8 levels from nothing to 20-fold normal. So 20,000 milliunits is 20 times the normal levels of factor 8. This was actually kind of an astonishing experiment. Even within hours, these dogs had attained normal levels, and the levels went up as high as 20 times normal. But you can see by days 6 to 10, the levels disappeared. That was accompanied by liver toxicity, hepatotoxicity. These are liver enzyme levels showing that liver cells are being lost in this time. There's a peak here early on, and then by 10 days, these levels of ALT are the sort of things you see with acute hepatitis. So these dogs had severe liver cell damage. And then the last thing you can see that happened is that both of the dogs, these are Haemophilia A dogs, developed factor VIII inhibitors. One of them went as high as 1,200 Bethesda units by 10 days. And the other dog had a Bethesda titer of about 36, 37. So this told us that this gene delivery vehicle, adenovirus, was profoundly inflammatory. And indeed, some of you know that a teenager, Jesse Gelsinger, died from an adenoviral therapy back in 1999. So this will not be used in hemophilia gene therapy. Absolutely, that's not going to happen. There are works going on now to look at lentiviral gene therapy. It's still in preclinical and early phase clinical development, but everything I'm going to discuss now is about AAV, or adeno-associated viral vectors. And it's really important to remember this is not adenovirus, it's adeno-associated virus. And these are two completely different beasts. So here's an adenovirus. On this electron micrograph, you can see pointed by this yellow arrow, and all these other much smaller particles are AAV particles, adeno-associated viral particles. They're about 20 nanometers in size. This is about 100 nanometers. So it's really critical you remember that differential, because I showed you what happens with adenovirus, and that doesn't happen, never occurs with AAV. So the advantages of AAV as a gene delivery vehicle is that the wild type virus, the normal AAV virus, doesn't cause disease in humans. There are various forms of the virus, different serotypes, which are the protein coat of the virus, and that allows you to deliver the vector to different tissues in the body. And then the other advantage, although we'll return to this at the end of my talk, is that the vast majority of the vector exists outside of the host chromosomes. So it's extra chromosomal, and it exists in what we call episomes within the nucleus, these so-called circular AAV vector polymers. And again, I'll return to this later in the talk. So you change a wild type virus genome, shown at the top of the slide here. So this has about just over or just under 5,000 nucleotides of single-stranded DNA. When you want to turn this into a gene therapy genome, a gene therapy, if you like, chromosome, you remove this yellow region, the so-called cap gene, and this region, the rep and the cap genes, and you insert the coding sequence for factor VIII or factor IX, depending on whether you're treating hemophilia A or hemophilia B, and you deliver this with a regulatory sequence, a so-called promoter and enhancer region, which turns on this gene in liver cells. So this is a part of the gene, the part of the transgene, which says, we want you to make factor VIII or factor IX in liver cells, hepatocytes. So you can see the only other parts of the viral genome that are left are these so-called inverted terminal repeats here and at the far end of the gene here. So this is about 10% of the viral genome. Now, you can insert into this cassette the coding sequence for factor 9 quite easily. 1.4 kilobases fits in here very easily, but you have to remove the B domain encoding part of factor 8 to get the factor 8 cDNA, complementary DNA, into these vectors. So this is actually what we call a packaging limitation, which is a bit of a concern, or at least it's an obstacle to doing hemophilia A gene therapy easily. And then this slide shows the final part of making an AAV vector. Here is the genome that I've just been describing to you. Here's the transgene. Here's the enhancer promoter element. You now then put this nucleic acid sequence into a protein coat. So you package this into its protein coat, the so-called particle capsid, and then you inject this into a peripheral vein. And depending upon the code that you use, as illustrated by these different colors, you can target different organs, different tissues within the body. And I've outlined in red here all of these vectors, all of these capsids, which will go to the liver. And I'm going to say some words for sure about AAV5, AAV6. And you can see that all of these have actually been used in clinical studies where you inject the vector into a vein, a peripheral vein, and the vector finds its way into the liver and ends up getting inside the liver cells, the hepatocytes. So about 15 years ago now, we treated 8 severe hemophilia A dogs with AAV, followed them up for over 10 years. Their previous untreated factor 8 levels were 0, and they bled about 4 or 5 times per year. And then over the 10 plus years, their levels after single injections were over 10%, and their bleeding events essentially disappeared. So this gives you an idea in a large animal model of over a decade of success and elimination of bleeding. And humans, the Nathwani study, which was back in 2011, now has human hemophilia B cases who are out to over 9 years following treatment. So now up to 2020, and this is the hemophilia A study from BioMarin using an AAV5 vector. And this is after 4 years of follow-up in 2 cohorts. In green, 7 individuals treated with a higher dose of this vector, and in orange, a group that's treated with 60% of the vector dose. And you can see that around about week 25, the levels of factor 8, which are shown over here on the vertical axis, were round about 70 or 80% for the mean levels. Some individuals had as high as 170%, and some individuals round about 10%. So lots of variability. And then a fall off after the first 2 years. So here's 2 years after delivery. And now at 4 years, the mean factor 8 level is 24%. The median is 16%. And here are these numbers in tabular form. Year 1, here are the mean values: 64, 36, 33, and currently around about 24%. And what did that do to the clinical outcomes? Here are the 4 years in the same study in these 7 individuals treated with the highest dose of AAV5. So this is the patients free from bleeds. So how many of the patients were not bleeding at all? Pre-infusion, only 14% of these patients were bleed-free. They were on prophylaxis. And then you can see somewhere between 71 and 86% of these patients had no bleeds after AAV gene delivery. And their annualized bleed rates went from over 16 down to these very small numbers between 0 and 1.3. So a marked improvement in their clinical outcomes. So for the rest of the talk, the last 12 or 13 minutes or so, I'm going to talk about the reliability, the variability, the durability, and the safety of gene therapy for hemophilia. And the first thing to point out that is, we believe that once an effective vector dose has been identified, and that will usually occur during these smaller phase 1-2 studies, usually involving somewhere around about 5 to 15 patients, we then have a good sense of the therapeutically relevant amount of transgene that needs to be given, and you will get a reliable response in more than 90% of cases. Not everyone will get a positive response, but the vast majority will show evidence of clotting factor production. But it is worth remembering on this slide here, here is the original Nathwani study in 2011 that I've referred to. He used an AAV8 vector, and here is the vector dose in vector genomes per body weight. You can see now that after up to the latest BioMarin study, the one that I was just referring to with factor 8, AAV5, we've now increased the vector dose 300-fold from these studies that were done back in 2011. And so there has been a very substantial vector dose, and there obviously hasn't been a 300-fold increase in factor levels. So this is some indication actually of the inefficiency of this process. I mean, this works. It's working very well in many patients now, but it's a relatively inefficient gene delivery process. Now, what about the variability? Well, I showed you on the BioMarin studies that at peak levels, some patients had 10% levels of factor VIII, and some were as high as 170%. So in the factor VIII trials, we've seen up to a 10-fold variability. I think we're now beginning to believe that factor IX studies are significantly less variable. And here's a couple of examples of this. So here at the top is one of the factor IX gene therapy studies. This is one of the Unicure studies. There are 3 individuals here, followed out to 52 weeks. This is with the Padua factor IX transgene. This is a hyperactive transgene. And you can see that 1 year, 52 weeks, here are their factor IX levels. They're anywhere between 31 and 50%. So they're pretty closely clustered. In contrast, in this study, a factor VIII study, the Sangamo Pfizer study, again going out to 1 year, you can see that the factor VIII levels go in from anywhere close to 150% down to around about 15, 20%. So a much higher variance here compared to the factor IX study. So why is that? So first of all, is it really true? And I'm not sure we absolutely know this for sure, but it looks as though it may play out this way. The first thing is that we're using liver cells to make the protein. That's where factor IX is normally made, but it's not where factor VIII is normally made. So that may be a bit of a problem for hepatocytes, for the liver cells. Are we putting those cells under some sort of stress, something we call the unfolded protein response, when they're making large amounts of factor VIII? Does that make the cells less comfortable, and therefore there's variability between patients. Factor VIII requires von Willebrand factor to carry it, and we all have different levels. Hemophiliacs have different levels of von Willebrand factor, and that may be altering the factor VIII levels to a varying extent between individuals. And then lastly, unfortunately, whatever you do with factor VIII is inherently more challenging. It's a bigger gene, it's a bigger protein, and the protein is more labile, more fragile. And so this is something that as clinicians and biomedical scientists, we just know is a fact of life. Now, I'm not going to spend a lot of time on detail with this slide here, but it illustrates to you that here is a very blown up version of a cell. Here is a vector in stage 1 being taken up by that cell, a liver cell. The vector then goes across the cytoplasm. It gets delivered into the nucleus here at phase 4. The DNA is released. You need a second strand of DNA produced here, and then some of the DNA gets integrated, but the majority stays in these episomal forms. The DNA is then made into messenger RNA. It's then made into protein and secreted. And the reason I show you this slide is that we really do not understand very well about many of these events. So there are 9 different processes here, and honestly, our basic understanding of these events is still fairly minimal, and it's likely that variances in these events between individual patients may explain some of the variability we see in gene therapy outcomes. What about durability? Well, I've already shown you data that in the canine model of haemophilia, we had over a decade of persistent expression. And this has also been shown by the group in Chapel Hill, Tim Nichols and Denise Sabatino. So I think in large animals treated once, 1 infusion of AAV vectors, you have good management for over a decade. And in human studies, the factor IX studies are now out to over 9 years. And in factor VIII, I showed you the 4-year data from the BioMarin study. So I think we can say in 2021 that we should be able to get at least 5% factor levels for at least 5 years. I think these numbers are actually pretty easy to defend with our current level of knowledge. So lastly, what about the safety of gene therapy? So first of all, following the infusions, there are infrequent and transient reactions to the infusion. And this is usually safe. Some patients may get some fevers, but allergic or other acute responses are very unusual. In the midterm, over the first 3 to 6 or 7 months, somewhere between 50 and 75% of patients develop some minor level of liver toxicity. This is usually transient, and it usually can respond to a course of steroids, which usually lasts somewhere between 1 and 3 months. So this is not uncommon. We don't really understand exactly what's causing this, but it usually goes away within the first 6 months following delivery. And then I'm going to finish by just saying a few words about longer term potential issues. So is there a long-term genotoxicity risk with AAV gene therapy? And genotoxicity means when you deliver these vectors, do you alter the host DNA? So is the patient who receives the gene therapy, is their intrinsic, their native DNA, their native genetic material, being altered by the vector. And at the bottom here, you see a very recent molecular therapy reference from Ken Cornetta, stating that AAV joins the rank of genotoxic vectors. So what does this mean? Well, when you give AAV vector doses that are currently being administered, I've already highlighted that you're giving many particles. So 10 to the 15 particles is, for those of you who are numerically challenged, which so I am too, this is some idea of how many particles you're giving. This is 10 to the 15 vector particles. Only a very, very small fraction of this gets into the host genome, much, much less than 1%. But much, much less than 1% of this is still a lot. And so when you inject the vectors, at least 10 million integration events will occur within the liver. And that's really a manifestation of just this huge number of particles that gets injected. And so very recently, the group in Chapel Hill and the group here in Kingston in Canada have been able to look at some of these details in the dog studies that were carried out over more than a decade. And we now know that the integration is non-random within the genomes of these animals. I should point out, and this is really, really important, that when we investigated these dogs, when they eventually were euthanized because they were suffering old age problems, on detailed examination at autopsy, there was no evidence of any liver cell tumors of any sort in the numbers of dogs here and in the other colony. This is a pictorial representation of the integrations in the UNC, the University of North Carolina dogs, that was published by Denise at the end of last year. These are the dog chromosomes. There are 38 of them and an X chromosome. And you can see that in these 6 dogs, there were integrations scattered throughout the genome. But when you look in more detail, as we did in the dogs here at Queen's, some of these integrations occur repeatedly, but they seem to be in safe areas, non-oncogenic, non-cancer producing regions of the DNA. So in the last couple of minutes, first of all, we're excluding current patient populations listed on this slide at the moment. If you have antibodies to AAV, which is the case in about 30 to 60% of the population, you're not eligible to enter one of these studies currently. There are no pediatric studies ongoing, and this is probably several years away. There is just beginning to be some interest in inhibitor patients being included in studies. And then finally, if you've failed or you've lost expression, which actually to this point hasn't happened in clinical studies yet, you would have developed antibodies to AAV, and you wouldn't be eligible for a retry of therapy. So at the moment, AAV therapy is a one-shot deal, although there may be ways around that, hopefully within the next 2 to 5 years. So this is my last slide. I've shown you, first of all, really that phase 1-2 clinical trial numbers, like the BioMarin study, are small, usually less than around about 10 patients. Clinical studies really do require about 2 years to mature sufficiently to assess the efficacy, how good, how effective the therapy is. Immune obstacles, that is pre-existing antibodies, and the possibility of redosing are still significant hurdles. I've just told you that if you lost expression after gene therapy, at the moment, you couldn't be retreated because of immunology, because of antibody development. And then, although the biological knowledge of transgene delivery, expression, and persistence is still limited, so I try to stress to you that there's still lots more we need to understand. None of these considerations should limit the licensing of a gene therapy product within the next couple of years. And so we anticipate, I think, reasonably that either this year or in 2022, there will be the first product licensed and available. And with that, I'm happy to stop and certainly very pleased to take any questions. And I think I'm happy to stay and chat with some of you after the session. So I'm going to stop sharing my screen. Thanks for your attention.